So hi everyone, welcome to Raaonline. This is Dr. Srivatsan, your faculty for biochemistry. And in today's session, we'll be seeing about gene expression in eukaryotes. Okay. In the previous session, the previous part, we have seen about the gene expression and regulation in prokaryotes. Okay. Since eukaryotic cells has got a larger DNA and this DNA is much more complex than when compared to that of the prokaryotes, the regulatory steps are much more in eukaryotes when compared to that of the prokaryotes. So we are going to cover this topic in two parts. The first one will be the expression, how the genes are being expressed in eukaryotes. And the second part of this will be how this is being regulated in eukaryotes. So we are going to see this in two parts, right? So what do you mean by gene expression and gene regulation? As I told you before during the time of the same topic in prokaryotes, that how come you have got all the cells in your body, all the cells are basically having the nucleus, and each of these nucleus has got the same DNA in it. Okay, so basically all the cells in your body has got same DNA in it. Why only some particular organ expresses some particular proteins which are not expressed in other organs? How come hydrochloric acid is synthesized only in your stomach but not in your lungs or not in your brain? Okay, so how is it possible? This is possible by regulating this huge DNA. Okay, so huge DNA, actually we have got 3 to 3.2 billion base pairs, okay, in the DNA, and out of this the coding region is only a meager 1.5%. So out of those 3 to 3.2 billion base pairs, you are going to code only 1.5% of the region. Okay, and even though it is just 1.5%, it also forms a considerable number. Clear? But the total number of proteins which can be seen, which is being guessed roughly in your body, is around 23,000. Again, there is a huge difference between the 1.5% and the amount of protein which is being encoded by your body. How this is being expressed in different kinds of organs, and if it has to get expressed at one organ and if it has to get repressed at other organ, then it has to undergo a tight regulation. Without any kind of a regulation, this kind of an expression or repression is not going to occur. Okay, so I'll first see what are the different kinds of ways by which the gene is being expressed in an eukaryotic DNA. So this is all about this topic. Okay, so gene expression in eukaryotes, it can be expressed in 4 of the following ways. Okay, by housekeeping gene or inducible gene. The second one is through the presence of enhancer or an inducer, as well as the silencer, which is otherwise called as a repressor. Okay, the third one will be about the cistrons. How the genes are being expressed in an eukaryote is through the different kinds of cistrons. Okay, the fourth one will be the interaction of your genetic material, that is your DNA, with that of the protein. Okay, so this is an entirely different universe and this protein is another entirely different universe, and these two are getting interacted. Okay, the purpose of this interaction is to express certain kind of things, okay, to regulate certain kind of things in DNA, because you need to have some very complex mechanism in order for regulating such a huge number of DNA. Okay, and it has to be foolproof, it has to get regulated at various levels, it has to get checked upon at various levels so that the mistakes would not happen. That is the basic concept here. Okay, so what are the 4 points? The 4 points are how the genes are being expressed in eukaryotes by the housekeeping genes or the inducible genes by the way of enhancers or silencers, by the way of cistrons and by the way of interaction between DNA and that of the proteins. Okay. Now, we will see in detail regarding each one of them and how it is helpful in expression of a gene in eukaryotes. The first one will be, of course, the housekeeping and inducible genes. So, what is this housekeeping gene? This housekeeping gene is otherwise called as constitutive gene expression. This is a constitutive gene expression. Okay. Whereas the other type is inducible gene expression. So, what is the difference of gene expression between these two, constitutive as well as the inducible? As the name suggests, the constitutive gene expression is the one in which there is going to be expression of this gene in a consistent basis or a continual basis. Almost all the time it is going to get expressed in the cell. Okay. So, these kind of genes are called as constitutive genes. Say for example, you take the enzymes of your tricarboxylic acid cycle, otherwise called as citric acid cycle or Krebs cycle. So, the Krebs cycle is extremely important for what? For the generation of ATP. High amount of ATP will be generated by your Krebs cycle, and this cycle is the basic cycle for the derivation, for the formation of different high amount of ATP. So, a cell which basically depends upon the glucose for its energy production, for the ATP production, will almost always need to have the genes for the enzymes of Krebs cycle to express every time, almost always. So, that this enzymes, the genes which are expressing the enzymes for this Krebs cycle will be synthesized all the time, and the Krebs cycle is not going to get stopped because of the absence of any kind of an enzyme. You get the point here? This cycle cannot be stopped in a normal cell, and in order for that cycle to go on and on and on, you need to have continuous supply of the enzymes of Krebs cycle. Okay, and in order for this continuous supply of these enzymes of Krebs cycle, you need to have the gene, and these genes has to continuously produce these kind of enzymes. So, these kind of genes are called as constitutive genes.